So today's seminar is a special one because we are lucky to welcome as presenters some of our very own students and postdoc. I'm excited today to be introducing Josh McInnes, McInnes, both our MSc students, our master students, Kristen Sora, a PhD candidate, as well as Dr. Harmony Martell, an Ocean Leaders postdoc. So the first talk will be by Josh. Josh is from Victoria and is currently enrolled as a master's student at the IOF under the supervision of Dr. Andrew Treitz. I enjoyed chatting with Josh over this corridor often, and it is very interesting to hear his stories about his research on killer whales. Josh's research focused on the ecology and behavior of marine mammals in BC and Monterey Bay, California, with studies focusing on the foraging behavior, diet, and ecology of transient killer whales and whistle dolphins. Today, he will be presenting on work he has undertaken around photo identifications and community structure of transient killer whales of California and Oregon.
Yeah, so thanks for having me today. I'm really excited. It's great to be able to share some of the studies that we've been conducting and some of the results from a recent study that's actually not so recent. It is over 10 years in the making, and the last 5 years have been kind of dedicated to trying to understand a little bit more about the killer whale presence off the outer coast of California and Oregon. So I'm a master's student in the Marine Mammal Research Unit with Dr. Andrew Treitz, and share some of the results with you. This study was quite a large study. It was with multiple different organizations along the coast and multiple people. And I was just lucky enough to have an interest and be able to lead it. So just to quickly give you an overview, photo identification involves a sort of mark recapture work where you take a photograph of an individual and you're able to repeatedly see that animal over time and you're able to distinguish it. And photo ID is well used throughout the marine mammal community. And you can see here, these are 3 different forms of killer whale that we have in the North Pacific. We have the residents, the transients, and the offshores. The residents are a fish-eating salmon specialist form. Most of us have heard of the southern residents of the northern resident killer whales, and they prefer neritic or coastal waters. We have the transients, which are a mammal hunting form that have large distributions. And then we also have the offshores, which are actually a fish-eating form, but they eat higher up on the food chain, and most of these larger species like sharks and skates. But each animal here can be distinguished based on congenital markings or markings caused by their environment. You can see scratches, shapes of the saddle patch, which is the gray area by the dorsal fin. You can also see notches. And these features stay with these animals throughout their lives and act as a way for tracking movements, association patterns between individuals, occurrences in regions. And it's a great tool that enables us to even do census work on populations as well as estimates. But our study was really looking at the transient killer whale population, which is an ecotype, and an ecotype is a term we call a genetically distinct group. And transients are actually split into multiple different populations. And we have in the yellow is Big Gulf of Alaska Bering Sea stock that goes from all the way from northern British Columbia all the way up into the Bering Sea and around the Aleutians. We have the West Coast transients, which are the population that I'm primarily concerned with in my thesis. That's around Southeast Alaska, all the way down to Southern California. We have this little genetically distinct population called the AT1s, which are currently on their way out into extinction. There's only 7 left but most of these populations don't intermix or associate. But what we're even starting to find, which really caught my interest, was, with most studies occurring with this coastal West Coast population since the 70s, was what's really happening south of Washington State, off the coast of California and Oregon, where a lot of information on killer whales has not been published or studied. Most of this work has been done in the coastal waters. So we formed a study where we used multiple different methods of data collection. We had opportunistic marine mammal surveys, primarily in Monterey, California. where we collected a lot of long-term behavioral data as well as photo ID work. We collected a lot of sightings information from whale watchers based on a citizen science approach, as well as fishermen, and then also dedicated survey work off NOAA ships in collaboration with National Marine Fisheries Service. Our study area encompassed the waters from basically Astoria, Oregon, down to Point Conception. So encompassing the whole entire Oregon coast down to the limits of Central California, with an extent out to 560 kilometers offshore, with most of that outer coast area being dedicated to line transect surveys. And one key factor here just to look at is the continental shelf, which is an important variable for finding killer whales and looking at the different assemblages or communities that we're currently interested in, is that how it narrows as you head down the coast from Oregon to California. So what we found was off Oregon, we're starting to learn that there's a couple of different assemblages of killer whales or communities within this West Coast population. Through the photo ID work and through looking at associations, we can kind of link together different groups based on how much time is spent in each area. What we found was that there's a bit of a coastal assemblage that spends their time mostly really close to the coast, within 3 or 4 kilometers, often entering small inlets. We have an oceanic group, which I'll talk a little bit about in a few slides, as well as an outer coast group that spends most of their time near the outer shelf. And this map right here just really shows the number of sightings off the Oregon coast, with 97 records, and the gray being the unknown, which most of the gray we believe to be based on photo quality. They are transient. We're just not sure which community they are. California, very similar. We had more sightings, so more encounters. We had up to 260, with most of it being in Monterey, which you can see here. This is Monterey Bay. It's this amazing ecological system full of deep water canyons. That are one major big canyon called the Monterey Submarine Canyon that stretches, basically bisects the middle of the bay from Moss Landing to far offshore. But most of the sightings occurred in this deep water, where the continental shelf comes right into the coast. During this time, we spent a lot of time working with the Moss Landing Marine Laboratories at San Jose State University. They provided 5 years of access to their library, their resources, to a bunch of volunteers and researchers. We sat there after our surveys on the water. We'd do 8 hours on the water, and then we'd do 8 hours in the lab. So by the end of the day, we were pretty tired. We did this for spring and summer for about 5 years straight. So our results from the study were that we found our study went from 2006 to 2018, and we encountered transient killer whales 146 times during surveys. And we analyzed over 113,000 photographs of individual whales. And we were, at the time, the US government, NOAA, was really interested in putting together a census because they didn't have a lot of information for killer whales south of Washington State. And so we took it upon ourselves to really try to put together a catalog of individuals to help their stock assessment reports. There was a total of 150 whales, though, that we did find that were commonly seen or between years, as well as 30 matrilineal groups from repeated sightings. When killer whales were identified, we gave them a special alphanumeric designation. So for instance, here you got the OCT system we developed, which just means outer coast transient, to distinguish these individuals from the coastal groups that we see. The 30 number would be the 30th animal that we encountered. And then as well, the B would be, say, the second offspring of OCT 30B, and then her subsequent offspring would be OCT 30B1. On most years, the number of years photo identified, we found that we saw a majority of the individuals at least on 1 year of identification, but some were more rare throughout the years that we see. So there are some animals that would show up only once in 10 years, which was quite shocking. So this really shows that our study is still needing more a rigorous approach, looking at mark recapture and more of and that we do not know the overall population. We found that each year we identified new whales, either through new whales being seen in the area as adults, or also seeing calves being born, and this was consistent throughout the study period. And we also were able to track individuals throughout their life. Here's an individual here in 2006, just before he was sprouting at around the age of 14. and we were able to track him throughout his life up until recent. Actually, we had a recent report of him. So we were able to build a life history of a lot of the different groups that were in off the outer coast of California. This is one matriline we know as the OCT30s, and you can see here the matriline system's based on a female descent. And you see the genealogy here between a mother, her presumed brother, and you can see her offspring right here as you head down this family tree. One of the interesting results from the study was that a potential for what we're calling an oceanic population that we're currently not sure where they belong. We've identified 40 different killer whales between 1997 and 2021 based on photographs. And many of these animals could not be linked through association or were never seen in association with any of the outer coast transients that we identified off Oregon or California. Most of these whales were found far offshore near seaward of the continental shelf, up to 300 kilometers. But on numerous occasions, they were seen predating marine mammals, similar to what the transient form feeds on. And we also noticed one interesting characteristic was the presence of cookie cutter bite marks or scars. You can see these circular patches here. They're quite nasty wounds that these little pelagic parasitic sharks leave on large vertebrates. And these sharks are only found in warm deep water ecosystems far from shore. So we're starting to find that there might be a population that lives far from sea, far off of the shore, that may be a new type of killer whale. We're still kind of looking into that. The results from this ended up being a new technical memorandum published through NOAA. This was the results from that study. It took a long time to really put together, but it came out this year, and we're pretty happy about it. It's kind of the first chapter of my thesis that I'm working on. The next steps will be really to look at the behavioral association patterns, occurrence patterns, and seasonality of some of these different communities, as well as looking at the fine scale spatial movements, potentially using satellite tagging. But yeah, thank you very much for listening to my talk.
Next, we have Max Miner, also our MSc student at IOF and working under the supervision of Dr. Brian Hunt and Dr. Camilla Speller. I communicate frequently with Max because of his role as co-president of the IOF Student Society. So I'm very excited to hear more about his research today. His work at the IOF focuses on using environmental DNA and Asian DNA methodologies to examine harmful algal bloom dynamics along the Pacific Northwest coast. Today, he will be presenting on the work he is undertaking for his thesis using DNA extracted from nearshore marine sediment core samples to reconstruct past harmful algal bloom events at a clam garden site in the traditional territory of the Gitga'at Nation.
Wonderful. Okay, so I'm going to be talking today about my thesis work, but before I do that, I want to make clear that my work is part of a much larger project. It is not something that is mine alone, but is the work of many researchers, many students, and most importantly, is a collaborative project with the Gitga'at First Nation. Then takes place, all of it, in their territory in northern coastal British Columbia on an island called Finn Island at a place known as Lax Kwil Dziidz, which is a Tsimshian term that means the place that squirts. It is named after the clams that grow there. And the Gitga'at have been harvesting clams there and managing these clam fisheries for thousands and thousands of years. So I want to make it clear that I'm going to be showing just a little part of a much larger story that belongs to many people. But because we're short on time, I'm going to be concise and start talking about my thesis work. I am doing my thesis on an issue known as harmful algal blooms. So harmful algal blooms are a catch-all term for anything that phytoplankton do in the ocean or in freshwater that people don't like. It might be that your dog drank out of a weird pond in the backyard and gets sick, or it might be that phytoplankton do what they do in the ocean, which is take sun and turn it into more phytoplankton. And in doing so, they can create toxins, which then accumulate in the tissue of marine invertebrates like clams, crabs, barnacles, etc. These toxins can then do harm to the people who eat these clams. Globally, we're seeing an increase in these harmful algal blooms through a variety of anthropogenic factors, changes to marine environments that are associated with human activity. Ocean acidification, warming oceans, nutrient input increases, et cetera, all contribute to a global increase and a regional increase in harmful algal bloom activity. This harmful algal bloom activity has a significant impact on people who eat clams from the marine environment. This image here are areas of the British Columbia coastline that are currently closed to butter clam harvest, including our study area in red. And the main takeaway that you should get from this slide is that it is all of them, and has been that way for quite a long time. However, the indigenous peoples of the British Columbian coast have been managing around these harmful algal blooms for thousands of years. So there's sort of this collision between our current day management practices and the way that they get in the way of sometimes the traditional management practices and subsistence fisheries of coastal communities and indigenous communities across British Columbia. My thesis work, as I mentioned, takes place on an island called Finn Island in the territory of the Gitga'at Shellfish Nation in northern coastal British Columbia. This site is a very important site to the Gitga'at. They have been living there, actively managing the clam population there for many thousands of years. There are various cultural pieces of traditional ecological knowledge and stories that are associated with the site. There is ongoing collaborative archaeological work on the site that has found dates going back 4,000 to 5,000 years. And that's not of people occasionally visiting, that is of people living there, of people transferring knowledge intergenerationally about how to take care of these clams. Unfortunately, over the last decade or so, the levels of harmful algal bloom toxins in those clams have been found by the Gitga'at's own monitoring efforts to be above the limits that are allowable for human consumption. So there has been this impediment of traditional harvesting practices at this site caused by harmful algal blooms. And our project broadly is looking at how these harmful algal blooms were managed in the past to inform how the Gitga'at can revitalize their fishery at Clamstown, and going forward, how to protect that food source for food security in a changing world. My role in that is to look at the past. Unfortunately, harmful algal blooms don't fossilize very well. You don't find a little phytoplankton fossil. They also don't preserve in the archaeological record. So we have to turn to different methods of looking at them. And the method that I will be using and am currently using is ancient DNA. Ancient DNA is just a catch-all term, again, for all of the old DNA around. And we're specifically working with ancient environmental DNA being all the DNA that's floating around in the air, your DNA floating around in the air around you, the DNA of whales and fish and clams and phytoplankton floating around in the water around them. As that DNA floats around, the clams, which are filter feeders, are sucking the water in, eating the organisms in it, and also trapping the DNA that is floating in the water in their shells. As they build their calcium carbonate shells, they fix DNA in that shell matrix. Similarly, the DNA falls with particulate matter out of the water column and piles up on the seafloor in sedimentary DNA. So we have two sort of parallel sources of ancient DNA that we're working with, these sediment cores from just offshore of the site and the clam shells from the archaeological village site at Clamstown. And we are using these proxies for biological events in the past to learn about the abundance and frequency of harmful algal bloom activity at this site specifically, and hopefully the province more broadly, to inform, again, the restoration of this site and the return of clam harvest to Clamstown. We are, for the good of the province and for the good of the Gitga'at Nation and anyone who likes eating clams, because if you've tried to harvest clams before, you know you got to check those toxin levels before you go out there. And that's it for me. I'm still in the process of data work, so maybe someday I'll have some more data to present to you. But thank you everyone for listening, and I'll be happy to answer questions later if anyone has.
Thank you so much, Max. That was great. We have time maybe for one very quick question before we transition over to Kristen. Andrea, go ahead. I'll unmute you. There you go.
Thanks so much. Great talk, Max. I'm curious, is this work part of the Indigenous Aquaculture Collaborative Network? Are you part of that group or linked up with them? And is Spencer Greening someone that you're working with at all in this work?
Yeah, tangentially. It is part of Dana Lepofsky's work with the Clam Garden Network, and through that is linked to the Indigenous Aquaculture Network. I have not worked directly with Spencer, but given that we are working in similar spheres, when I was up doing field work, I got the opportunity to have dinner with him and chat about the different work that we are both doing. So I'm familiar with his work, but we aren't directly collaborating on this specific project at the moment. But yeah. And
awesome, great to hear, and happy to facilitate a connection if ever that's useful.
Thank you so much. And to speak to what Charles said in the chat, I want to, affirm that the map that I showed was not a strict claim of territory, but an area of resource usage that in areas where the Gitga'at have in the past and continue to use resources. These maps are obviously a complex issue, and it wasn't meant to be a statement of them owning that particular chunk of land exclusively.
Kristen Sora is a PhD candidate working under my supervision. Kristen is also well known among us as the social lead for the student body. So our supervisory meetings often intermix with science discussions and ideas about IOF social events, making it quite fun to conduct. Kristen's research topic encompasses multi-stressor impacts on the Canadian Beaufort Sea marine ecosystem at various biological scales. Today, she will be telling us about how she has developed and applied food web models to understand system dynamics in the Beaufort Sea ecosystem in the face of change, with a view to inform the management of an important marine protected area site in the area.
Today, I'll be discussing the Beaufort Sea historical changes and its relevance to management. For a quick overview, first I'll discuss my thesis and the study area, which is the Beaufort Sea and the TNMPA. And then I'll go into the ecopath with ecosim model that I'll be using throughout my thesis. So my thesis framework includes how changing ocean conditions have altered the structure and function of the Beaufort Sea shelf and protected areas, using ecopath with ecosim as a tool to investigate. The first chapter includes increased sea surface temperature and reduced sea ice extent, with future chapters including data updates to improve the data quality and reduce uncertainty, the addition of multiple climatic stressors and their interactions, and future projections to the end of the century. I'm also collaborating with Amanda, Milang, and Emma and IRES for their mercury simulations within the Beaufort Sea. But today I'll be discussing my first chapter, as this has been submitted for publication in the Arctic Science Special Issue. The Beaufort Sea is one of 5 priority areas for integrated management under the Oceans Act, which means DFO is responsible for a precautionary approach to sustainable use, development, and protection of areas and resources, and incorporates social, cultural, and economic values. It encompasses the marine portion of the Nunavut settlement region, here in purple, and includes diverse ecosystems, such as the Mackenzie River, the Beaufort Gyre, and the Beaufort Shelf, which we'll be discussing today. The Beaufort Shelf ecopath with ecosim model covers a shelf area of 103,000 kilometers squared, and stretches from the shore to the 200-meter contour line. Conditions for life on the Beaufort model are determined by ice cover and sea surface temperature. For example, extreme low ice years have coincided with decreases in body condition, productivity, and growth rates. Within the study area is the TNMPA, and it's Canada's first Arctic MPA. It has 3 sub-areas that have been traditionally used by the Nunavut and is important culturally and economically. The conservation objective is to conserve and protect beluga whales, anadromous fish, waterfowl, and seabirds, their habitat, and their supporting ecosystem. Priority indicators that form the framework for monitoring and assessing the NPA are ecosystem structure, so that's the individuals and communities, the ecosystem function, that's the energy flows, and population structure and health of key species. For anyone who's not familiar with ecopath with ecosim, I'll do a quick overview. So ecopath with ecosim is a widely used software for describing the structure of ecosystems, provides outputs at the ecosystem level, reflects food web linkages, energy cycling, and changes in biomass of ecological functional groups, which defined here are single species or groups of similar species. Shown here is the ecopath flow diagram from producers to fisheries. So this is a static snapshot of the mass balance trophic model. The size of the circles are the relative biomass of each of our functional groups, and this is at the beginning of the time series. And the gray lines are the connections of functional groups to each other. The model contains 31 living functional groups, 2 detrital groups, and 7 subsistence fisheries and harvest groups. We have our key species here, so it's beluga whales, bowhead whales, ring seals, and Arctic and polar cods. EcoSim, on the other hand, is a time dynamic model that builds upon the initial ecopath model. It is fit to time series and is used to address ecosystem-based management questions. Shown here is a change in relative biomass of functional groups from 1970 to 2012. The model simulates changes in the food web over time under climate forcing, and that's represented by increased sea surface temperature and reduced sea ice extent. To avoid biases triggered by short-term extreme events in this historical model, 5-year averages were used at the beginning and the end of the time series. We also have plugins such as ecological indicators, which includes the trophic level, diversity, and biomass, and ecological network analyses, which is the energy flows and cycling. And these allow for additional analyses on the environmental status of our modeled ecosystem. And the priority indicators for management were synthesized with these plugins to describe key trophic interactions and energy flows, with a focus again on our key species, beluga whales and Arctic and polar cods, and to quantify the ecosystem structure and function with changes in climate. The Beaufort Sea shelf beginning and end of time series averages were compared to 4 other ecosystem models. This is the Gulf of Alaska, Eastern Bering Sea, Eastern Chukchi Sea, and the Barents Sea. And they were chosen for this analysis as they're located at similar high latitudes, are mostly less than 200 meters in depth, seasonally ice covered, and are driven by subsistence harvests and commercial fisheries. So for our results, from 1970 to 2012, the Beaufort Sea shelf temporal structure was relatively stable. From our ecological indicators plugin, we have trophic levels, so the marine mammals and birds trophic levels were like our neighboring ecosystems, where many linked ecosystems will actually be sharing the same migratory marine mammals and birds. The fish and benthos trophic levels were comparable across all ecosystems, whereas the zooplankton trophic levels were similar to our distant eastern Arctic, which is the Barents Sea ecosystem. Our diversity, as seen in the Shannon Diversity Index, was relatively stable, and our total ecosystem biomass increased. However, it was only 20 to 55% of the total biomass observed in all the other ecosystems. Over the time series, the majority of our total ecosystem biomass in the Beaufort Sea shelf was held in lower trophic levels. So 90% of the biomass in the model was at trophic level 2.7 and below. And this is similar to the Eastern Chukchi Sea and the Barents Sea. However, the majority of this biomass was contained in primary producers and zooplankton, whereas our other ecosystems, so that's the Eastern Chukchi Sea, the Barents Sea, and the Eastern Bering Sea, held it in benthos, whereas the Gulf of Alaska was evenly distributed. From our ecological network analysis indicators, we had an increase in total system throughput, and so that's the sum of our ecosystem flows, a stable fin cycling index, and that's the amount of total system throughput cycled. But the other high latitude ecosystem models had flow values that were 2 to 4 times that of the Beaufort Sea shelf, which indicates that there were fewer connections and pathways. Arctic and polar cods were consistently ranked first in Keystone due to their low proportion of biomass and significant impacts on the ecosystem. Arctic cod plays a substantial role in the Beaufort Sea shelf ecosystem, transferring energy from primary producers up the food web to beluga whales. And we have differences here in our key species versus those outlined in management. And that's because our study evaluates ecosystem linkages and energy flows. And it's highlighting the species which are key to sustain the overall ecosystem health that supports the culturally important species outlined in management. So for our big takeaways, the Beaufort Sea shelf ecological indicators and ecological network analysis indicates temporal ecosystem stability, even with increased sea surface temperature and decreased sea ice extent, suggesting that ecosystem function was not largely affected by climate changes over our historical time period. However, the Beaufort Sea shelf had less flow, cycling, and total biomass than the other ecosystems, indicating a smaller ecological size with fewer connections and pathways. Compared to the other ecosystem models, the Beaufort Sea shelf has a distinct structure from strong primary and secondary producer influences. And Arctic and polar cods can provide insights on ecosystem changes occurring within the TN MPA and the surrounding Beaufort Sea shelf, and provide valuable information on the effectiveness of the MPA management and the achievement of conservation objectives in a changing climate. Thank you.
Last but certainly not least, we will hear from Dr. Harmony Martell, an Ocean Leaders postdoc at the IOF, working with Dr. Simon Donner. Her research links fine-scale mechanism of coral physiology, thermal performances, and stress memory to large-scale phenomena, such as coral bleaching events. Harmony is currently developing models to better understand the mechanisms of coral bleaching using the relationship between light, temperature, and metabolism. Besides being a researcher, I got to know that Harmony is also a seasoned educator with lots of experience teaching science at the high school and university levels.
Okay, great. Thanks for having me this morning. Just to give you a little bit of background before I dig into this, since I was little, I've been pretty pumped about corals. At age 11, I saw them with my very own eyes for the first time. And at age 20, I sort of took a pilgrimage to Australia. I spent a semester during uni there, and I got my scuba certification on the GBR. And then I came home after that to realize, home being the US, to realize that we were doing a really poor job of managing coral reefs, in my opinion. And that was also the first time I saw bleaching. And so I wanted to make better tools to help manage them. So in my master's, I developed biooptical models to non-invasively quantify bleaching, but the field was still moving too slow, in my opinion. And corals have persisted on the planet for hundreds of thousands of years, having survived at least 5 mass extinction events. So surely they've evolved these mechanisms to cope with stress. And so the thought was in the 80s was that, acclimatization was going to be like a sufficient thing, like corals would be fine. And people started to sound the alarm, and it was actually met with a lot of disagreement and a lot of conflict in the coral community. And so we started to sort of talk about, like the royal we of coral people, started to talk about this concept of coral thermal tolerance, which is defined as the ability to withstand big deviations in sea surface temperature. And so some work I did in my PhD early on showed that even though some corals can acclimatize to thermal stress, not all of them can. And I really wanted my children, oops, I think there's a slide missing here. There we go, sorry. I really wanted my kids to be able to see reefs the way I have, not only in aquaria. So I needed an intervention strategy. And so in the final PhD chapter that I did before I came to UBC, I really started to dig into this idea of creating more resistant corals. I didn't feel like I had the time to wait around for some long-term intervention, and coral growth is relatively slow. And at the time, getting corals to reproduce in the laboratory was very hard. Now we've sort of unlocked the secrets for coral spawning, and we can get it to work in the lab pretty well, but that's only been in the last few years. So when I began this work, there was some evidence in the coral literature that the corals had the capacity to acquire thermal tolerance, though it hadn't been expressly quantified. And I had read about this concept of stress memory in the broader literature. And so this concept of acquiring stress tolerance is certainly not a novel idea. Rather, it's evident across a wide variety of taxa, from bacteria to humans. And in the broader literature, this highly conserved response is referred to as stress memory rather than acquired thermal tolerance. And so the way it sort of works under this particular model, can you see my cursor? Okay. If you imagine what these authors of this review call a triggering stress, this black line here would be the fitness of an organism, exposed to that stress would be reduced over time. But if the if the organism is previously exposed to what these authors called a priming stimulus, you might expect this blue trajectory here, where there's an initial cost to the organism, but when affronted with that same triggering stress, you would see some benefit from that. And so you can think about this as sort of like a training or putting the organism on a treadmill. You'd expect that there'd be a fitness benefit for that. And so in this trained scenario, the organisms would outperform the naive organisms. So of course, in my PhD, I didn't have, this was sort of a risky project and I didn't have explicit funding. So I applied for a bunch of grants, some really big, some really small, and I got none of them. But I really wanted to do this project. So I made a video and did some crowdfunding. And I mentioned that because I'm happy to chat with anyone about that that might be interested. And I strongly advocate for students to consider that as a viable funding source if you need funding. So before I can tell you how to train corals, I need to tell you how the heck we quantify bleaching. And so what we do is we take parts of the tissue and separate it from the skeleton. And this can be done a variety of ways. Airbrushing is my favorite. We collect it into a tube. We mix it all up to separate the coral parts and the algae parts. And then we quantify the algae and relate it or normalize it to the surface area. And this gives us a measure of bleaching per unit area, or the more algae that there are, the less bleached that the coral is. So if we think about the model that I just showed you, to train corals, we might expect something like this. If we have a bleaching event, we would expect a reduction in coral pigments or algal density. But if the corals were trained, then we might expect them to do better. So there would be some cost. Sorry, my slides are going slow. Whoops. Some cost, but some perceived benefit after a bleaching event. And so a lot of this was a crapshoot, to be honest. I was working with a species I hadn't worked with before. I had no idea what dose would work to train my corals. I was looking for a Goldilocks zone. So too little, I'd see no training. Too much, I'd see bleaching. So I really needed something just right. So one of my committee members said, you have to do as many treatments as you can. And so I managed to run 6 treatments, which damn near killed me. So here is a picture of me after my corals were still alive when I planted them in the field. Here is a picture of me after my 3rd night of sleeping in the lab on the floor. And here is a picture of me, oops. Here's a picture of me after the entire experiment ran. So here's some of the results. It was clear that the amount of training mattered. So I looked at the doses of the actual training. And so training was definitely dose dependent. So on the x-axis here is degree heating days, which is a measure of thermal stress. And on the y-axis is algal density. So this is a measure of bleaching. And so what we see is that the lower the thermal stress, the higher the density, and the higher the thermal stress, the lower the density, which means greater bleaching. So it seems pretty linear. That not really a big surprise there. And so there was somewhat of a cost to that training, and the cost was linear related to the dose of stress. But here are the interesting results if we look at all of the treatments over time. And I didn't have time to include this slide, but just believe me that each of the doses of priming were slightly greater until there was a dose that was really strong, and I'll show you that in a second. So here are the controls. They didn't change much despite being poked and prodded for this entire 2 weeks. And the naive nubbins, which are the ones that were not trained at all, bleached. So that's what we expected them to do. And the strongest training treatment bleached my corals right away. So that was too hot. But my other 3 training treatments seem to have some slight costs, but an initial benefit. So they reduced bleaching, roughly 32 to 51% reduction in bleaching. Okay, great. So we can train corals to reduce bleaching in the lab, but who cares? So this is what brought me to UBC. Now I'm in the climate and coastal ecology lab working with Simon Donner and students and other amazing people in his lab. And so I wanted to know if training is something that's even a thing. Like, is that even happening on reefs everywhere? And so we actually know there's been one study that has looked at this in the Great Barrier Reef. And so they've looked at what they call this sort of protective trend, where there's a protective period. So this would be a training period, and then a recovery period, and then a bleaching event. And what they found is that that's actually the predominant thing that they see on the Great Barrier Reef. So that's interesting, but what about the rest of the world? And is it actually effective? And so if it is, should we be protecting those areas where it's effective? And so Simon has amassed this amazing curated global bleaching database. And so we'll actually be using a newer version of this database that is being written up right now in the lab. And again, this is a great thing so this is one of the things that I'll be working on actually collaboratively with another student, with a student in the lab, a PhD student. So we're really excited to find out sort of what this looks like. And again, this is great. My results of my study are great, but it's one species at one point in time in the lab. So what is that dose relationship with bleaching and thermal stress? And so there are tons of studies, in the coral literature that use a range of thermal stress doses. Why don't I amass those and see if how dose gives or modulates the shape of this response curve? And so this is just a snapshot of this huge database that we're populating right now with the help of other students in the lab. That is going to look at the dose responses from the literature and the relative doses of thermal stress to try to tease out the shape of that curve. But what actually happened is I had jumped a few steps ahead. And so if you think about stress memory, it means you've gotten the dose right and there's a benefit. But actually, there's this idea of hormesis, which is the biphasic dose response, where low doses of stress may be beneficial, but high doses are deleterious. And so since I had so many treatments in my stress memory experiment, I had a range of doses. So if you consider the doses here, training intensity, and look at the response, what you see is that no training results in bleaching, but some training results in this bump, this benefit. And then too much training, again, results in a problem. So bleaching is actually hormetic, and this is the first time that people have looked at it in this context. There's some evidence that hormesis actually is heritable. These benefits are heritable in the coral literature, but that's just come out in the last year. And so since we found this is exciting, but these hormesis-mediated phenotypes are not that much. That's like 30 to 60% benefit. And that's in line with what we know in the literature for other organisms. And so to pursue this a bit more, I've been awarded a grant in collaboration with some folks at FIU in Florida to investigate the mechanisms of these hormesis. And we think there might be an epigenetic component. So we're going to do a really large experiment with a bunch of doses and actually outplant some corals that'll be used in restoration. And so we want to see if we can train corals for use in restoration. And so that's work that's ongoing. So yeah, we think that this priming or training may elicit a beneficial response. And so in summary, we can train corals to resist bleaching. Truthfully, we have some preliminary evidence that it's already happening in some reefs around the world. And we think we'll find that it does happen. But we also think that we may have exceeded the amount of warming that corals can adjust to, given the increasing frequency and duration of marine heat waves. So this isn't a silver bullet. And really what we probably need to do is reduce emissions to save coral reefs and the species that they support, like our species. And so if I have any time, I'd be happy to take your questions.
Go ahead, Zach.
Yes, can you hear me? Yep. I was just wondering, in terms of like the effort required and the success rate, how does this compare against artificial insemination for the coral?
That's a good question. So they're two different things. And this is the idea for this to be used in restoration would be paired with coral reproduction efforts and selective breeding. So I think that we don't know the answer to that question yet. This concept of stress memory, and what we know about it and hormesis in the literature, is that the hormesis mediated phenotype, resilient phenotypes, can be relatively short-lived. So when we would expect it, if we measure this in the environment, we might expect the benefit to last somewhere between a week or a little bit longer. So that's not very useful, right? But there's a couple ways that could be beneficial. A lot of evidence in model organisms has shown that this phenotype can be extended up to a few months. And so if this is something that we could possibly dial in, then the idea is that we could train corals to survive at particularly vulnerable times, either in their adult life or in actually younger life stages. And so that could be beneficial. And then the other thing is, we do have a little bit of preliminary evidence as well that corals that have been trained, the parents seem to give some of that, if it's while they're breeding, that benefit seems to be heritable. And so the idea would be to do all three, to train corals, use the trained corals that are the strongest for selective breeding efforts, and also the outplanted corals, prime them or train them for these stressful times that they might see to maybe benefit restoration yields. So that's sort of the idea. It would be a multifaceted approach. Did that answer your question?
Yeah, I did. Thank you.
No worries.
And I think with that, I want to thank our four speakers, Josh, Max, Kristen, and Harmony, for your amazing talks. It's always so great to hear the diversity and so many interesting topics that our students and postdoc are working on at our institute.